Manshaei. I'm a senior research associate in SynLab Toronto. Happy to present one of our projects, Tangible Chromatin, here in ISS conference. Digital technologies have had a significant impact on exploring and analyzing of experimental datasets in the field of biology by providing interactive tools. While many tools exist for exploring DNA sequencing data and gene regulatory data like GeneMania and BioGrid, an area that presents new challenges for tool design is the analysis of the data from high content microscopy experiments. These experiments can generate giga or terabyte of the high resolution images, which are often computationally processed to detect key information, but the algorithms are error prone, requiring manual inspection to validate the algorithms, which leads to fewer samples analyzed. Moreover, the data is often aggregated, which is useful to understand population wide statistics but can miss low frequency but biologically important events. Another challenge is the lack of methods for biologists to track and monitor unique events and to find and assess the impact of pattern variation. Lastly, the current approaches in this area don't allow for collaboration, exploration, and interpretation, even though the data is typically analyzed by teams of researchers working together. We respond to these challenges by building on a growing body of the work and that uses tangible and embodied interaction techniques paired with large interactive displays to support data analysis and exploration. Our goal is not to replace the existing algorithm approaches used by biologists like ImageA, cell profiler, but rather to complement them with interfaces that can visualize the detailed information and results from the image processing algorithms, provide interactive approaches for browsing, selecting, and comparing individual data elements, expand options for productive collaboration through both independent and joint work. Our design process involves a collaborative team of HCI researchers and biologists working together in an iterative manner. We design around the datasets of our biology team members who use chromatin fiber microscopy to study how cells replicate their DNA. Briefly, and their approach captures high resolution images of chromatin fiber, I mean DNA strands with proteins attached. These are captured under different conditions, for example, with drugs or without, called wild type. This data is analyzed to understand how abnormalities in DNA replication regions called forks can cause cellular damage and evolve into cancer. We analyzed we designed a tangible chromatin, a tangible and multi-surface system that extend existing chromatin fiber data pre-processing techniques, allowing users to monitor patterns in segmented data and outside of raw data images. We followed an iterative process to design, develop, test, and define the system components. These were decided on as a group by our interdisciplinary team and then implemented by the CI researchers and tested by the biologists in an ongoing manner. We met on a weekly basis to assess progress, make design decisions, and determine next steps. Tangible chromatin includes three main components: an interactive tabletop display 48-inch by 81 inches, a large interactive wall display 48-inch by 324-inch, and one or more mobile phones that serve as active tangibles. We use tangible and multi-touch interactions on the tabletop for browsing and selecting data elements across multiple experimental conditions and pair these with a broad, especially explicit overview of the selected data on the wall. To use their own mobile phone with the system, a user places it into a cradle with a fiducial marker affixed to the bottom. On the tabletop, selected groups of fibers are displayed around the mobile phone, with each fiber is displayed in a condensed form in order to provide an overview of the respective percentages of each of the region types. Since fibers can differ a lot in length, users have the option to normalize the length of fibers on the table. This makes it easier for users to visually compare the percentages of the different regions between fibers. Individual fibers can be highlighted by rotating the mobile device and then selected by confirming on the mobile device's screen. When selected, a fiber is displayed on the wall underneath any currently displayed fibers. On the wall display, selected fibers are visualized in their full extent in order to show the layout of all the regions as well as the locations where the proteins bind. As fibers differ in length and some can be very long, zooming on tabletop allows users to view specific regions of a fiber up close on the table while retaining the full fiber view on the wall. By selecting the zoom option, the mobile device is used to cycle through fibers on the wall and select one, which is then shown on the tabletop. Swiping and two fingers gestures are then used to pan and zoom the fiber on the tabletop. Each user's data navigation mobile device is assigned to unique color, which is reflected in the circular menus that appear around it, as well as on the area of the wall where their selected fibers are displayed. If two or more users are working at the table, each with their own mobile device, the wall display will be divided into corresponding colored regions, and each user will manipulate the fibers in their own region. However, since the wall display is large and visible to all, users can discuss the data and share observation with each other anytime. Users may sometimes want to compare individual analysis more closely. Therefore, we design a flexible data integration mode to transition between individual and merge analysis. When a mobile device is in data integration mode, a shaded circular area appears around the device, defining a space on the table where the data from multiple data navigation devices can be merged. We conducted a formal user study that investigated how tangible chromatin supports biologists in individual and collaborative data analysis tasks of data sets from microscopy imaging of chromatin fibers. Our specific goals were to understand how participants use the system to generate ideas both individually and collaboratively, and how the way participants interact with the system both independently and jointly impacts their analysis. We recruited participants with a biology background and baseline familiarity with molecular and cell biology principles so that they had the necessary domain knowledge to understand data and tasks. Eight biologists, three female, five male, all aged 18 to 45, participated in the study. They were invited to come in randomly assigned pairs as the study involved both individual and collaborative tasks. Our biology team members created a realistic but synthetic data set to provide users with a task scenario that was manageable within this time frame of a user study. Since chromatin data is complex and researchers typically need to spend time exploring it on their own before discussing the collaborators, participants were given free related tasks for this study. For task 1, an individual data exploration and analysis task, each user was asked to analyze a chromatin fiber dataset. This task was designed to evaluate the participants' ability to use the system to recognize patterns and find irregularities in the data. Half of the user one from each team were given fibers from an experimental condition in which the cells had disease causing mutations called mutated, while the other half, user 2 from each team, were given fibers from an experimental condition with normal cells called wild type. Tasks 2 and 3, two collaborative data interpretation tasks. For task 2, the two users were asked to work together to compare the fibers from two conditions and discuss the differences. This task was designed to evaluate the user's ability to use the system to collaboratively share the patterns discovered in task 1 and compare them to identify differences between the two data sets. For task 3, two users were asked to examine three fibers from an unknown condition and jointly decide if they were all mutated or wild type fibers based on the patterns they had noticed in the previous tasks. This task was designed to test the effectiveness of the collaborative sense making from task 2. The study began with a pre test questionnaire to understand participant background experience. I mean, use of data analysis visualization tools, familiarity with chromatin fiber data, if how they collaborate in data exploration. This was followed by a brief system demonstration. Participants were asked to use their own mobile device, and extra mobile devices were also available. Participants were then given practice time to get familiar with the system, and an experimenter was available to help as needed. The practice lasted on average 11 minutes. After these, participants were given task 1 followed by tasks 2 and 3 with as much as they needed to complete each task. Finally, participants completed a post-task questionnaire covering functionality, learnability, user experience, and usefulness of our design system. We draw an concept of pragmatic and epistemic actions from Anto, Resch, and Maglio to analyze user interactions. Steve et al. Have suggested that classifying the actions users make with a system as either epistemic or pragmatic can help in understanding how well the system supports problem solving. Epistemic actions help users reduce cognitive load through alternations in their world that lead to a decrease in task complexity and play an important role in problem solving tasks. In contrast, pragmatic actions are definitive and directly bring the user closer to their goal. We coded all interactions with the system as well as gestures and verbal interactions as either pragmatic or epistemic based on these prior works. We also coded the number and value of the ideas users had through during the task where an idea is one or more observations made with the system. This figure illustrates the results of epistemic pragmatic coding of task 1 and task 2 and 3. All users started with a series of pragmatic events and actions to place fibers on the wall, as this was required to begin their exploration analysis. The sequence of interactions varied by user, but we found that all users except user 2 of team 2 performed at least some epistemic events and or actions in their process. Similar sequences of interactions can be seen between users of each team. In task 1, particularly for teams 1, 3, and 4, the similarity between the sequences followed by team members, even though they were working independently, may be attributed to the fact that they were working at the same system at the same time, so possibly took inspiration from each other in deciding which step to take. It's interesting to see how different and team 2 looks, which can be explained by the fact that the task 1 user 2 of team 2 didn't do any epistemic actions, and then in task 2 and 3 team 2 users had to talk a whole lot more compared to other teams, because it was user 1 of team 2 having to explain to user 2 of team 2 more about data. The top row shows the uniqueness of the idea generated by each user in task 1 and task 2 and 3. The darkest portion of the bars shows ideas that are unique to the user. The lighter portions of the bars show the ideas that were common between users in the same fiber condition and common within or between teams. The bottom row shows the value of the ideas generated by each user in task 1, task 2, and 3. Darker colors indicate higher value ideas. As teams progress through the tasks, the number of high valued ideas increase. The notable exception to this is user 2 of team 2, who, as described above, had to spend more time getting familiar with the visualized data compared to task 1, task 2, and 3 had more of a high valued ideas. At the end, Tangible Chromatin allowed high-value idea-generating observation of patterns that would otherwise be lost to aggregate averaging. There was some indication that the support for epistemic actions were helpful in generating unique or valuable ideas. This is consistent with research and that has studied how epistemic actions reduce cognitive load in problem solving tasks. Our work tasks a step in this direction. However, further study is needed to understand the relationship between epistemic actions and ideas, as well as to understand how different kinds of interactions can support users in moving from observation to ideas to interpretations in the analysis of the complex data. We structured the study tasks as individual data exploration followed by collaborative interpretation based on the existing practices of our biology collaborators, who frequently shift between individual and group work. They spend time getting acquainted with the data on their own, then bring it on the table for group discussion and interpretation. Tangible Chromatin effectively supported participants in moving from individual to collaborative activities. Working individually, almost all participants engage in both pragmatic and epistemic interactions with the system. As a collaborative, though, teams shifted to using more communicative interactions than direct interactions with the system. They also shifted from quantitative data to qualitative and comparative statements. We saw some indication that epistemic actions may be important for generating valuable ideas and that this also impacts their ability to perform effectively when they transition to joint work. I want to use this opportunity and thank all team members for their effort on this project and all funding agencies, NSERC, CRC, CFI, MRI, which have supported this project over several years. Finally, thank you all for paying attention to this presentation.